Good evening, guys, and today our topic is about the apoptosis. And in the previous lecture, we have discussed about the reversible and irreversible cellular injury and necrosis. So let's get into it. First of all, what is apoptosis? In contrast to necrosis, apoptosis is a programmed cell death, and it is physiological. But necrosis is pathological, and necrosis does not require any energy. It is not an active mechanism, but apoptosis is energy dependent. It means that it is ATP dependent. What are some examples of apoptosis? The menstruation, first of all. In during the menstruation, our endometrium sheds monthly, so it's an example of apoptosis due to hormonal changes, and it is physiological among the women. And in embryogenesis, removal of cells, for example, formation of the anal canal and formation of fingers, formation of digits. These are some examples of apoptosis. In case if apoptosis does not occur as it was programmed, so it leads to some mutations. For example, syndactyly. Syndactyly means that some of the digits are fused. It can be of hands and foot. As you can see in the diagram, there is a fusion between the digits of the hand and foot. These are some mutations related to the apoptosis. Okay. The third example of apoptosis is the CD8 positive T cell mediated killing of the virally infected cells. What does it mean? It means that if a cell is infected by viruses, then these cells present the viral peptides by the MHC class 1 on their surfaces. So CD8 positive T cells, T lymphocytes, they recognizes the virally infected cells, and they mediate apoptosis among them. How does this process occurs? First of all, CD8 positive T cell virus kill the virus infected cell by they recognizes the small pieces of viral proteins or viral peptides, which are displayed on the surface of the infected cells using MHC class 1 molecules. What are MHC class 1 molecules? These are major histocompatibility complex class 1. It means that those cells which are infected by viruses, they express or they present viral peptides by using this molecule MHC class 1 on their surfaces, and when they represent the viral peptides, so CD8 positive T cells recognizes them as virally infected cells, and they mediate apoptosis among them. Okay, what is the morphology of apoptosis? Morphology of apoptosis is different from necrosis because necrosis has various types, and according to that types there are different morphological appearance. For example, liquefactive or colligative necrosis, coagulation and gangrene, etc. But in case of apoptosis, it leads to the cell shrinking. The cell shrinks and it leads to the cytoplasm become more eosinophilic, means that it became dark pink in color. Also, as the nucleus is concerned, the nucleus become condensed. It results in pycnosis. This is the medical term for nucleus condensation. And the nuclear fragmentation is known as the karyopexis, means the nucleus breaks down into small pieces or fragments. After that, there are apoptotic bodies which are formed as a result of apoptosis, and these bodies are removed by macrophages. Okay, and this apoptosis does not followed by inflammation, as in case of necrosis. After the necrosis there are chances of inflammation, and there's always inflammatory process going on, but in case of apoptosis it does not lead to inflammation. Okay, as you can see in the diagram, this eosinophilic cell is the apoptotic cell, the cell which is undergoing apoptosis. So you can see here in the diagram. Okay. What is the mechanism of apoptosis? Like some brief mechanism, I can explain you that it's mediated by caspases. How a cell know that I am going to die or I am going to undergo apoptosis? It is due to the caspase activation, and caspase activates the proteases and endonucleases and other enzymes that results in the cell breakdown. Caspases mostly 90% activates proteases. Proteases are enzymes that break down proteins. As you all know that our cell cytoskeleton is made up of proteins like tubulins. Okay, so these are broken down by proteases, and caspase also activate another enzyme known as endonucleases, and these endonucleases break down our DNA and RNA. So these caspases activates further enzymes that break down the cells, and eventually the apoptosis occurs. Okay, what are the mechanisms of apoptosis? By which mechanisms apoptosis can occur, or by which mechanisms the caspases can be activated? So there are two mechanisms by which caspases are activated, but we will discuss three of them. Okay. First of all, the most important and the most popular is the intrinsic mitochondrial pathway, and the second is the extrinsic receptor ligand pathway, and third we have discussed a bit just before, it's cytotoxic CD8 positive T cell mediated pathway. So let's discuss them one by one. First is the intrinsic mitochondrial pathway. Intrinsic mitochondrial pathway occurs in case of cellular injury. Okay, if the cell is injured or DNA is damaged, or it leads to hormonal stimulation. These three mechanisms, first is the cellular injury, DNA damage, and loss of hormonal stimulation. These three mechanisms, if occurring inside a cell or either of three, it leads to the inactivation of BCL2. What is BCL2? Let's skip these two lines and focus on these uh, apoptotic factors. Okay, there are two kinds of apoptotic factors. First is the pro-apoptotic factors. It means that they are influencing or stimulating the apoptosis. The second is the anti-apoptotic factors, which are inhibiting the apoptosis. Pro-apoptotic factors are BAC, BID, and BIM. There are a lot of them, but I have written just four for you for easily understanding. It's BAC, BID, and BIM. These are pro-apoptotic factors, like they are favoring the apoptosis, while anti-apoptotic factors are BCLXL family and BCL2 families. These are anti-apoptotic factors. So, coming back, the loss of hormonal stimulation or DNA damage or cellular injury leads to inactivation of BCL2. BCL2 was protecting the cell from apoptosis, but when its inactivation occurs, so it leads to apoptosis. Okay, lack of BCL2, it leads to the leakage of cytochrome C from the mitochondria. As you all know that cytochrome C is a molecule that is present in the inner mitochondrial membrane, and when it is guarded by the BCL2, but when inactivation of BCL2 occurs, it results in the leakage of cytochrome C, and when the cytochrome C is leaked into the cytoplasm of the cell, it activates the caspases, and we are discussing nothing but caspase activation processes, right? So it leads to the caspase activation. Okay, coming to the diagram, this is the intrinsic mitochondrial pathway. For example, anti-apoptotic proteins, these are inhibited, right? Anti-apoptotic proteins are inhibited. So pro-apoptotic proteins, or BACs and BAC, these are pro-apoptotic, and BID is also pro-apoptotic. So these are activated. So It results in the permeabilization, okay, increasing permeability of the mitochondrial membranes. It results in the leakage of cytochrome C from the inner mitochondrial membrane, and it causes the activation of caspases, which further activates the other caspases, which are effector caspases, caspase 3 and caspase 9. Eventually it leads to the apoptosis of a particular cell. Coming towards the second mechanism of caspase activation, it's extrinsic receptor ligand pathway. How it occurs? First, let's come to the diagram. In the extrinsic pathway, there is a ligand. Okay, the ligand is binding to the receptor. Okay, the ligand can be Fas ligand, it can be tumor necrosis factor. It binds to the Fas death receptor. It's Fas death receptor, and when it binds to the Fas death receptor, it like directly activates the caspases. How it's occurring? The Fas ligand or tumor necrosis factor, TNF, binds to the death receptors. Death receptors can be Fas death receptor like CD95, or it can be tumor necrosis factor receptor. Both of these ligands, either of them binds to the receptor, it results in the activation of caspases. And in case of the extrinsic pathway, mostly caspase number 8 is activated, and in case of intrinsic, first caspase 9 is activated, and it results in the activation of effector caspases, which are 3 and 7. What are some examples of extrinsic receptor ligand pathway? It's the negative selection of thymocytes in the thymus. As you all know that thymocytes, which are thymocytes, thymocytes means that those leukocytes, T cells, T lymphocytes are activated in the thymus. They undergoes differentiation, but in the thymus there are two mechanisms by which these lymphocytes undergoes differentiation. First is the positive selection, and the second is the negative selection. Among these two selections, the negative selection is the example of apoptosis. By which mechanism? By extrinsic receptor ligand pathway. In the positive selection, it's just for an extra information. In the positive selection, the T cells with functional T cell receptors that recognizes self-MHC molecules survive. What does it mean? It means that those T lymphocytes that recognizes self-MHC molecules, major histocompatibility complex molecules, these T cells survive. But in case of negative selection, what kind of T cells are eliminated? Those T cells are eliminated that bind strongly to the self antigens. It means that those T cells which recognizes our own cells as antigens. It means that if those T cells are not removed immediately, it leads to the problems of autoimmunity, autoimmune disorders, for example lupus, for example rheumatoid arthritis. So these T cells have to be removed, and these are removed by apoptosis. Just let's revise these two like diagrams. Intrinsic, in which anti-apoptotic proteins are inhibited, pro-apoptotic proteins are activated. It leads increased permeabilization of the mitochondrial membrane. Cytochrome C comes out into the cytoplasm, activates the caspases. Okay, and in case of extrinsic, ligand binds to the receptor. There are two kinds of ligands. It can be Fas ligand, and it can be tumor necrosis factor ligand. It binds to the receptors, either the Fas receptor or it can be tumor necrosis factor receptor. So it leads to activation of caspases. Mostly in case of extrinsic, caspase 8 is activated, and in case of intrinsic, caspase 9 is activated, and they both activate the effector caspases, which are 3 and 7, and the results in the apoptosis. What is the mechanism by which the virally infected cells are destroyed by the cytotoxic CD8 positive T cells? As I have told you that, for example, this is a cell which is infected, which was originally infected by some kind of virus. Okay, we don't know what kind of virus, but just take an example that it has some virally peptides. These viral peptides are expressed by the MHC class 1 on the surface of the infected cell. These are expressed, okay? When a cytotoxic CD8 positive T cell is coming through it, coming like just passing by, it recognizes these MHC class 1 molecules which are representing the viral peptides. So it recognizes that this cell is virally infected and it must be removed from the body. It starts to mediate apoptosis. How it mediates apoptosis? It has like CD8 positive T cells releases perforins and granzymes. What are these two enzymes? Perforins creates hole in the cell membrane of the target cell. Okay, you can remember by perforations. So it creates holes in the cell membrane of the infected cell. And granzyme is also released by CD8 positive T cells. It enters the pores, through the pores, and activates the caspases, and eventually when the caspases are activated, apoptosis is triggered automatically. I hope you understand these mechanisms. Two are important: intrinsic and extrinsic, and CD8 positive T cell mediated pathway you can also remember as an additional information. This was for today, and in the next lecture we'll discuss more about like growth adaptations, cellular injury, and cell death. It's the part 4, and in the next lecture we'll discuss more about it. So thanks for watching, and I will see you in the next video. Till